Okay, hello everyone. Probably some more people will come in the next minutes, but I will just briefly introduce the topic for today so that we have enough time to cover everything. So today we are concluding our third iteration for our Monarch seminar series with focus on variant prioritization. 2 months ago, I think Yasmin introduced us to PhEval for benchmarking variant prioritization tools, especially those that are using or driven by phenotype-based approaches. And last time we had an introduction to variant prioritization in general. And today we are going to go a bit more in more depth into how to create your own kind of like variant prioritization evaluation component for PhEval. So if you have your own tool and you want to compare it to other tools, and Yasmin Bridges from Queen Mary University will go show us around that. So Yasmin, thank you very much for doing this, and take it away from here. And thanks everyone else for being here.
Thank you. Okay. Can you see the slides?
Yeah, looks good.
Yep.
So in my first talk, 2 months ago, we went over the architecture of PhEval, so how it's set out in these three broad phases, which is the data preparation phase, which we went through, the runner phase, and the analysis phase. So in the first talk, we went more in depth in the analysis phase, how we did the benchmark, and that was considering that we already had runners set up and that we could execute to get these results. So today, we're going to be focusing more on this runner phase here. So this is this runner execution where we run these variant or gene prioritization tools to get this format we need to do the benchmarking. So what exactly are we plugging in? Well, in the previous talk to this, done by Letizia, she went over variant prioritization tools and how they may be set up and how they process data and what the outputs are. So that's what we would be plugging in. So you start off really with PhEval focuses on tools that can consume this phenotype data. So these are HPO terms that can be represented in a phenopacket. And this always gets input into the tools to help with this prioritization for the prioritized genes. And then some tools, the variant prioritization tools, they also take as an input VCFs, which has the patient's genomic information. So this is just to put into the concept that this is what we're going to be plugging into that runner phase. So why build this custom runner? Well, first of all, you can integrate your own tool into a standardized benchmarking framework. So there's already numerous benchmarks out there published in different journals, and they all tend to show and evaluate different types of metrics. So with this way, with PhEval, you always get the same set of metrics that are evaluated, where we try and give the full picture, where it's not just focused on precision or it's not just focused on recall. It gives the full picture of how the tool performs. And this can help facilitate reproducible evaluation without modifying your core algorithm. So the key point here is that you just need to figure out how to interface your tool with PhEval. You don't need to rewrite your tool for it. And then this will also help when there are other groups out in the research that want to also benchmark your tool or see how it works. They can do it with the set out default way or the recommended settings that you want to highlight within your custom runner. So today I've done a few kind of pre-done videos on how to implement a runner, and I thought I'd just focus on a relatively simple one, which is Phentogene. This just does phenotype prioritization, so it doesn't look at the VCFs or the genomic data, but it gives quite a good and simple way on how you can implement your own runner for that. I've just added a link to the GitHub as well, so you can see how the tool works, and then the link to the paper. So generally, when I start out with writing a runner, there are three critical questions that I look at that tend to always correspond with the three different phases of the runner. So the three different phases of the runner is a prepare stage, a run stage, and the post-processing stage. So for the prepare stage, you need to look at is there anything that I need to pre-process before I actually run the tool? So what do I actually need to do before I want to run Pentagene? So with Pentagene, the only thing really that you need to do is you need to install the actual tool and then follow the recommended instructions. So the reason I don't implement this in the runner per se is because we don't want to have, say you want to run Pentagene on 5 different corpuses or 5 different configurations of running Pentagene, 5 different ways. You don't want to have 5 different installations of a large database for the tool to run. So we do this outside of PhEval, just as a pre-setup, and we pass that in as an argument instead. How do you run the tool? This is corresponds with the run stage. So what do you need? So Pentagene, it can't process phenopackets as is, but it can take in the HPO IDs just on the command line. And obviously we need the Python executable file from Pentagene as well, which is involved in the installation. And then what are the outputs of the tool? This is for the post-processing. We need to know what it looks like and how we can transform this into a Polars data frame for PhEval. So Pentagene outputs just a TSV formatted list of prioritized genes, and this is just a gene symbol and gene identifier and a score. So getting in how to set up your runner, there's just 3 prerequisites that you need to create a PhEval runner, and this is Python 3.10 and above. This is just for compatibility with PhEval. This is the minimum version that it needs. You also need Craft. This is for creating the runner from the cookie cutter template. So this is a official PhEval runner template that we have, and it helps just organize the project in a manner that would help with separation of code and also fills in some of the admin bits that can be a bit tricky if you don't configure it correctly. Then you also need poetry because the cookie cutter template, it creates a poetry project and we use this just to manage dependencies, and it also helps with the plugging features that we have within PhEval. So with this first video, it's just going into how we create this cookie cutter project from the cookie cutter template. So it's a simple command of craft create, and then it's the link to the PhEval runner template, and it will ask you 12 questions basically that you need to fill out on the command line. So you give the name of the project. So I've just called this Pentagene PhEval runner. The GitHub org name, so this would usually be the organization, but I've just done it under my username because this is for filling out the docs link. You can give a project description. This will fill out the README. And then there are some defaults as well that you can just use for this. So just use my Python 3.10 and the main file name will be main.py. I've given a name for the runner, which is Pentagene PhEval runner, and then filled in the author information. And then the rest I've just used defaults, and it takes in the tokens that you would use, and this would fill out the GitHub workflows. And you can see I've got this new repo, Pentagene PhEval runner. And we can see how this kind of looks like as a tree structure within the directory. And you'll see this brief kind of layout where we'll see we've got all of the kind of admin stuff that you'd have with a GitHub repo, how the docs are generated, and then this source is how, we have to prepare, post-process, and run different directories that would align with the 3 different phases of the runner stage. So how this looks in your IDE once you've set it all up, you can see we've got the full kind of project setup. We've got the docs link set up, the README has been filled in with the project description. We have all of the GitHub workflows that you need for a project that have the tokens input. So this will help when you want to deploy your docs and you want to publish to PyPy. This PhEval run directory, this is not created by PhEval. This is just something I've put in afterwards, but this is why I have the Pentagene installation in here, which is what I'm pointing at here, where I've put the name of the tool and the version. The Pentagene directory is just how we've installed it the default way, and that's the executable that we need to use. This config.yaml This is something that we need in every input directory that we pass to PhEval on the command line. And this just helps with specifying what tool we're using, the version, and what type of analysis we're doing. So Fentagene, it can only do gene analysis, so we just set that to true. And the tool configuration options, that's an optional field that we can input. So if your tool has additional settings that can be like twiddle a bit and to help with performance, you can add those there, and then those can be consumed within the runner to set these options while it's running. This corporate directory, I'm just doing a simple small test on one pheno packet, but we pass this to PhEval on the command line, and the results directory, that's where I'm going to put the results from the PhEval command. And then when we look in this Ventagene PhEval runner, so where all our code will go, we've got the post, process, prepare, and run. This is separated into 3 directories just to help with separating code. We have this CLI.py, which will just have a simple command that's input just to test if everything's installed correctly when we run the command. And it should just print hello world. And then in the runner.py, this is key. This is where we have that Fentagene PhEval runner. So this is the runner name that we provided on the command line. And this is where you will need to call all of your code within the prepare, run, post process, and it will run whatever you implement in this section with the PhEval run command. So how this plugs into PhEval, we provide this runner name as a path to the runner class within the plugin section. So you can see here we have this tool, poetry plugins, PhEval.plugins, and we have this name, Fentagene PhEval runner. And we provide the full path to the runner, and this is how it will interface with the PhEval run command. So what's going on in the background is that we are recognizing that this runner can be used by the main PhEval package that implements the PhEval runner class. And what's key here is that this is very much case sensitive. So if there is something slightly wrong, or there's one of the letters are the wrong way, then it will come up with an error that it can't find the plugin. So it needs to be case sensitive and match the full path. So with Poetry, you can specify which Python version to use. So here I've just specified Python 3.11, and this will create this virtual environment with Python 3.11. And then you can activate it using this eval Poetry and use, and it was use this will activate this environment so that you can install all of the dependencies that are specified within the PyProject.toml. And this will install the full projects. So this will allow you to run all the commands. So then if I was to run Ventagene PhEval runner, which should run the CLI the main command, it should just run, you see that you got the run command there. And as soon as you run that, you should get the hello world print statement. And then you can check if the Fentagene PhEval runner, if that corresponds correctly, that it would run these print statements within the terminal. And this just checks whether you have configured everything properly within the PyProject.toml. But this is, again, is handled by the the cookie cutter setup. So when you run the cookie cutter command, it will fill in that part in the PyProject.toml, and we just did that so that it would help with reducing any kind of issues that we get. So here I'm just running the PhEval runner command. I've set the input directory just to where it is because we have a config.yaml there. And I've set the runner name to Fentagene PhEval runner, and this will correspond with the class name, it will just be lowercase. I've set an output directory. That's where all the results would go, but obviously nothing will actually run apart from the prepare statements. And then the test data directory will just be where I have my pheno packets. And sometimes this can take a little while to run, I think, just as it all configures in the background the first time. And then we can check in this PhEval run directory, we should get some output within the results. So this can take a while, but you can see here as well that when the command where we have the input there, the runner, the output, and the test data directory, this all corresponds with the different arguments that we have within the runner command, within the runner class. So you can see that it did finally run. We've got some log statements that are done automatically by PhEval when you call certain commands, and then we have to prepare, running, and post-processing. So we can see that when we actually run this command and specify the runner that we've created, it does actually create all of the outputs. So we have this results.yaml that we use. So it just specifies what tool was used and the tool version if you supply it on the command line. The timestamp and also which corpus was used to run it and the configuration, which will just be a path to the kind of the input directory you supply on the command line, which would be the phentogene. So when it comes to actually preparing anything for phentogene, there is nothing really to prepare. So I just put in here nothing to prepare and modified the statement, and the output of the run should update with that. And then with the run, again, the things that you need to consider is what are the inputs of the phentogene, like what do you need? So here I'm just trying to seeing what the command is with the phentogene. So what do we need to actually supply the Python executable? Here I ran into an issue. This was just because I hadn't actually followed the default installation instructions of phentogene, but I fixed it obviously here and tried again. And here you can see here's all the default kind of arguments that we have with the phentogene. So we need to take, notice of what do we need to actually provide. So we provide, the HPIDs as -m, separated by an empty space. We need to provide a output, a path to where to put the outputs as because we don't want to use the default. And how do we name the file as well? So those are really the three things that we need to make sure we include in the command, as well as the database. So we need to specify where it can find that database that's used to be able to execute phentogene. So here I just specify a data class. I'm just gonna make sure it's clear what the arguments are. So it can be called in kind of in a clearer way further on into the code. So we need the executable, which here we're going to assume that as long as everyone has installed the phentogene the default way, it's always going to be found in the same place, which is in this phentogene directory, and it's going to be called phentogene.py. And then we specify the output directory, so where we're going to tell phentogene to output the results, the name of the file, and then the HPIDs, which will just be a list of strings that we're going to extract from the pheno packet. So within PhEval, there's a bunch of kind of helper methods that we've implemented that can be used by developers, and a lot of these are for extracting certain information from a pheno packet and reading in a pheno packet in the correct way. So we have this just like a pheno packet reader method that will read in a pheno packet as the protobuf object, which helps extract things a lot easier. And then we have, different methods to extract. So some tools may also want the age or The sex of the proband, so there's different methods within this phenopacket util class that we can use within on the phenopacket to retrieve this information. So here we're just taking the observed phenotypic features from the phenopacket, as phentogene can't handle excluded terms. And we just want the ID of these, and we're returning it as a list. So here I'm going into this class and I'm just showing you it can retrieve certain information, like it can retrieve a sample ID, all of the phenotypic features, the observed phenotypic features, or just the negated, and then just a bunch of other stuff that you may need and might be helpful within your code. So here I'm just going to specify this function. So with PhEval, we want everything to be traceable. So we've tried to help a lot with the metadata that will come up with each run, but also here we're trying to encourage that we're going to save the command that was actually used to run phentogene. So for each sample, you would have the command that was run or that will be run with phentogene, so that if you just wanted to look back so you can remind yourself what exactly was used on the to execute it, you can. So here it will just be we're writing to a command file, which will be stored in the output directory. Um, Basically all of these arguments, the executable, the output directory, the sample, to see how everything was run. And then this will be consumed again later within the PhEval pipeline to actually run everything. So we can, write this command, and we know that's just a Python executable. So we specify Python 3 and run it on the executable file. And then we specify, in the manual, all the HPO IDs that are related to that sample. And then we can specify the directory of where to store the files. As if you use the default, which would just be the current directory with a new directory called out, PhEval wouldn't be able to find that later on to do the post-processing. And then the name, and this should correspond with the phenopacket name so that we can match the output to the phenopacket. And then the data directory as well, just to make sure that we eliminate any confusion that could happen further down the line. So then we have this method here the idea is it would write a single command for a single sample. And this can be used alone to run phentogene. So then I think the final command would be just to write all of the commands for a whole corpus. And when I say corpus, I mean all of the phenopackets that we have contained within a directory that we want to run phentogene on. So for this, we would need the phenopacket directory, so where everything is stored. So this would be within the test data directory. And what's important is that the developer can you can define however wherever these files should be stored. So it could just be within the data directory itself, or it could be within a subdirectory named phenopackets. But what's key is that whatever you specify as the default, it should be outlined within the README so that it can be replicated by other people. And here, the phentogene executable and the command file path and the output path and the data directories it would be all of the same for the same corpus because we're running it on the same configuration. So this all files, this is just another PhEval kind of help command, and it will just get all of the files within a specific directory. So we can say for each file in this packet directory, we're going to write this command to this single file. And then this single file, as you'll see later on, will be used to run phentogene itself. And here I'm just defining for each packet what the arguments would be to run it. Obviously, I'm checking back to make sure I put everything in there. And I want the name of the file to be just the first portion of the phenopacket path to make sure that it will align later on with the post-processing where it will look to match the two. And then obviously we write the command for the sample itself at the end. And then I always find it helps to write the doc strings in either before or after, preferably before, just so you know that, or that I know exactly what I need to do in each of these functions and how they differ to the methods that we call it before. So now I create this final command, which would be to prepare all the commands. And this will call the command just above. However, we're going to be using the CLI arguments that we provide to the runner. So the input directory, the test data directory, the output directory from here. So the input directory is where you would find the Phentogene executable and where you would find the data directory. So here it is kind of hard coded, but again, this is just a very simple tutorial to begin with. These can be kind of manipulated and made flexible within that config.yaml that you put within the input directory. So then we have this raw results directory. So this will be the output directory. This is where we're going to tell Phentogene to output the raw results. The tool input commands directory is where this kind of command file is going to be written. So what commands do we want to run with PhEval and how with Phentogene and how we're going to run it. And again, the test data directory is just where we have our corpus of pheno packets. So here I'm saying that the Phentogene executable can be found within this Phentogene directory here at the side, and then within that directory it's called phentogene.py. So it is hard coded, but following the instructions, I mean, everything can be tailored to however the developer thinks best. So it can be hard coded so that users are forced to follow the default instructions for installing your tool, or it can be made flexible. PhEval can handle both and provide that both. Going to write the command file to the tool input commands directory and call it phentogene commands.txt, and then the output directory would just be the raw results directory. And again, this data directory where we're calling input directory.join path, this is where the Phentogene kind of data dependencies are. So where its knowledge base is, which is kind of always under this subdirectory called lib when you install it the default way. And then for the pheno packet directory, I've specified that is going to be in a subdirectory called pheno packets, as I've set up within that PhEval run kind of test directory. And then we can call this method within the runner. So within this run step, we call prepare commands. We import the method and we give it the same arguments here. So we have the self.input directory, test data directory within the runner. The raw results directory is kind of hidden, but you can access it from the runner class because this is built up within the the PhEval directory set up when you specify the output directory. So then the idea is that when you rerun this PhEval run command now, you will have to update the input directory here that to specify to point to the location of where that Phentogene default installation is, where I've got it within this PhEval run directory under the Phentogene. The runner name would be the same as it corresponds with the runner class. And then the output directory is wherever you want to output the results. I've kept it separated here, but within the same PhEval run subdirectory. And the test data directory will just be where I've got that kind of parent directory to the phenopackets directory. And I've also specified the version just so you can see how that end results.yaml will get filled up with the version of the tool. And you can see it's relatively fast. All it's doing is writing that one command to the text file. We've got all you can see that nothing to prepare statement is updated, and this passing phenopacket shows us that we've actually opened the phenopacket and looked in it, which is where we've called that command to extract the HPIDs. You can see we've got this single Python command to run the phentogene on these HPIDs that have been extracted from the phenopacket, and we've specified everything that we need to actually run the command. So then we need this kind of final run command. So we're going to run phentogene on this file, and it's just relatively simple, just Python, just using subprocess to run the actual thing. So for this, all you really need is the path to the phentogene commands.txt. So here I'm just iterating over every file that would be in that tool input commands directory, which should only be this one file called phentogene commands, which would be all the commands for the one corpus. And we run this subprocess and with the command file. We can just run it as bash because it has the Python 3 commands. And then again, this run phentogene method, we were going to call it within the runner class. So this run phase of the runner, all of this does is it prepares the commands and then it runs the tool on those commands that you've prepared. And we just give it this tool input commands directory that we can access from the runner class itself. And then we can run the PhEval run command again, and we should hopefully see that we'll also get some actual results from phentogene as well. And then you'll see some of these logs will be from PhEval, some of it will be from the runner, and then some of it will be from the actual tool itself. So this finished output path, this is all from phentogene. So you can see that something's executed and it's specified that the output path of the results should be within the raw results. And we can check this and check the format. So you can see, phentogene, it outputs this ranked prioritized gene list. So it has a gene and then the ID and then its score that is provided to it. And it's just basically a long list of genes. So turning this into the PhEval output should be relatively easy as it already kind of has everything that we need. And we can take note of how the file is named itself to make sure that the stem part will match the name of the phenopacket. So then we've kind of done the prepare, nothing to do in the prepare phase. The run phase is done, and then we move into the post process. So you have to think of it as the input of the post process phase will be everything that's in the raw results directory. So we need to transform that list of genes into this Polars data frame, where we've tried to recommend or tried to request just the limited amount of information so that it can be tailored to, can be accommodated by every tool. So it's just a gene name and a gene identifier, and then the score. We ignore the rank because we want everything to be ranked in the same way. So here I just specify a super simple method to just read in the pentagene result as a polars data frame. And we use polars because it's a lot more lightweight and faster than pandas, as it uses Rust as a backend. So you should only really need one kind of main method for this, which would be to actually extract the information that we need from the results. So something to note is that with PhEval, a lot of the gene identifiers are in the, well, we recommend for it to be converted into the ensemble format. So we have another helper method called this gene identifier updater, which can do a number of things. It can update the gene symbol, or it can get the gene symbol from an identifier, or get an identifier from the gene symbol. And this is what we'll use here just to find the ensemble ID for the identifier that's given. And then output of this is just going to be another polars data frame that we can feed into to PhEval. This is all described a lot more in detail in the docs, but within the data frame that you need to pass to PhEval, the columns need to be named a certain way. So we see that I will be renaming the columns to align with what PhEval will expect within the post-processing. And this is kind of the helper method that all runners will need to kind of to call within the pheno packet within the post processing, which is to generate the relevant result. And here you can see the schema. So this is how the columns need to be named. So here I'm calling this method from the gene identifier updater class, which will do to find the identifier for a given gene symbol. So it will call within this column to this method, and then you specify the return type, which will be a string, and you name the column, and it'll be named gene identifier. And then the final column will be the score. And we're just making sure that this will be classed as a float value. This just helps if there's any null values within that's output by the tool itself. Mm. So just moving along, we have this final method, which would be to create the PhEval results. So here what we'll need to do is basically for every raw result that's been generated by the tool, whether it's one, whether it's 100, whether it's 1,000, we need to go through each of those, extract the gene results, and then pass that along to PhEval with that helper command. And what PhEval will do, it will handle all of the kind of the admin bits of where to put the results and how everything needs to be formatted in the file format for benchmarking, which we went through in the first kind of part of this seminar series. So this output directory that you would provide here, which is the same, exactly the same one that will be provided on the command line, it needs the test data directory. So this would be where the pheno packets are, and this is just so within the post-processing stage, PhEval will label what the actual causative gene, variant, or disease is with a true positive label, and this helps with the benchmarking process. So this just creates that gene identifier class that will be used, and it just creates an instance of it first outside of this loop, otherwise it can become quite long. I actually made a mistake here when I specified the gene identifier as ensemble. It should be ensemble underscore ID. So you'll see that there'll be a bunch of errors in the console. So in this generate gene result, this is that PhEval helper method that's been defined within PhEval. And you provide it the extracted results. And then the sort order, this basically just tells it how the scores should be ranked. So whether it should be the highest scores are the best or the lowest scores are the best. It just tells you in what order these scores should be ranked. And then we can call this final method to complete the kind of process of PhEval, the three phases within the post process. Provide all of the arguments that it needs that we can access from the runner class. And then you can try run the pipeline again, but remove the previous results just to check that everything is running as I would expect. So these are the kind of warning messages that you get. They will say they could not find ensemble for the gene symbol, but really this was just because I provided the wrong string argument within that gene identifier class. And as you can see, the phentogene, it does just provide, I think, a list of every protein coding gene that it could possibly be within that output, which is why you see with a lot of these tools, it will always find the gene where it could be at rank 1000 or rank 1, but it will always tend to find the gene. And I run it again just to double check that was the issue. And you can see now, obviously it doesn't find it for some of them, but it does find it for the majority. And then you can see you've got more log messages of writing the results. It reads the pheno packet again just to label it. And you can see within this PhEval gene results, you can see it's found the right one at rank 1. We've labeled it as a true positive, and the rest obviously are not true positives. And these Parquet files are then what are consumed within the benchmarking, which I would have output. And then you obviously still get the raw results and everything else. So that's basically how you can set up a very simple runner. Obviously, there's a lot of things that are hard coded, but PhEval has the means to make it more flexible for your needs. But I just wanted to show you how things could be implemented within the runner. So we have all of these docs under the PhEval GitHub, the link. So this will just show you all of the different things that we offer for developing a plugin to make your life easier and how things can be done. And then I just want to say thank you to all of these people. PhEval wouldn't be what it is without everyone that's involved. And thank you all for your time.